how do you deconstruct a polymer that has been designed by nature to be resistant to deconstruction? This is a problem that our group and many other lignin scientists have grappled with for years. In trees, such as poplar, lignin is found in cells that have thickened secondary cell walls, and it is enmeshed with the cellulose and hemicelluloses in the cell wall and acts as a glue to hold that cell wall together. It provides rigidity and recalcitrance to the cell wall, which is incredibly important for providing structural support for the plant body, allowing long-distance water transport, and also acting as a barrier against pests and pathogens. As beneficial as this lignin recalcitrance is to the plant body, it is disadvantageous to many industrial processes which require the separation of lignin from cellulose and hemicelluloses to make downstream products. For this reason, one of the goals of our lab is to find ways to redesign the lignin polymer so that we can improve cell wall deconstruction without negatively impacting plant growth or development. In order to do this, we have to understand lignin structure and how best to manipulate it. If you look at this model of a poplar lignin that I have here, you can see that there are a variety of phenolic monomers that make up the polymer and also a variety of bonds that form between them. This is because the formation of lignin is chemically controlled, not enzymatically controlled. In theory, any phenolic monomer that's produced by a lignifying cell can get into the cell wall, can be oxidized by a lactase or peroxidase, and is compatible with the chemical combinatorial mechanism of lignin polymerization, is capable of being integrated into the lignin polymer. This introduces an incredible amount of plasticity or flexibility into the lignin structure. And we can use that to our advantage to rationally design a structure of lignin that meets our needs. In our case, we are trying to introduce more chemically labile bonds into the lignin backbone. The most prevalent bond you'll find in the lignin backbone in most native lignins is the ether bond, which is considered the most labile, even though it requires really high temperatures and really strong acids or bases for cleavage. We want to instead introduce ester bonds into the lignin backbone, which can break under mild base pretreatment at room temperature, thereby fragmenting the lignin and increasing accessibility to underlying polysaccharides. Because of the ease in which you can break these bonds, we refer to these ester bonds as zippers in the lignin and refer to the lignin itself as a zip lignin. We were very excited to find that some plant species, including poplar, natively produce zip lignin precursors that have ester bonds and that are compatible with lignification. However, they are produced in very low abundance. My research focuses on identifying acyltransferases in poplar that are responsible for making these zip lignin precursors, and so far we have found 8 candidates, and we are working to increase the amount of enzyme that is functioning in the plant to produce higher levels of zip lignin and to improve cell wall deconstruction. Thank you.
Thank you.